today I'm going to talk about contemporary evolutions and whether we can track the genetic basis of contemporary evolution in spatially heterogeneous environment. So as mentioned by Virginie yesterday morning during her introduction, but also by other speakers, so clearly now we can see evolution in action, contemporary evolutions in animals, different animal species and plant species. And this can occur over several decades, but even a few years. And but actually there is not so many studies reporting the genetic basis across genomes associated with this contemporary adaptive evolution. And if we want to predict so this contemporary evolution, actually we need to take into account also the role of the variability at the macro scale. This is especially true for the plant because the plant they cannot move. So here this is just a cartoon to explain that, for example, here you have a transect of 100 meters, and even it's very well known for like decades that actually the soil type can largely differ over the scale of very few meters. And also, so this is here I represent the species A, that also the species A will interact with different other species, like plant species, so species B and species C, sorry. So the general question that I have during my talk is what is the genetics underlying contemporary adaptive responses in population experiences, especially environmental variation. And to address these questions, so in my group we adopted three approaches. The first one is to set up experimental evolution in ecologically relevant conditions. The second one is to set up a resurrection study. And the last one is to monitor the demographies, the phenotype, and the genomic of the natural population. So today I will very focus on the two first approaches. Okay, so the model that we'll use, I will talk about is Abderrahman Taliana. So yeah, I'm pretty sure that you were scared that there would be no talk on Taliana today, but don't be scared anymore, there is a talk on Taliana. So you can see actually what I would like to show is that Taliana actually can live in different very contrasted habitats, included actually Taliana can live in a very highly competitive environment. So yesterday Magnus mentioned that maybe Taliana is the worst competitor weeds, but I kind of disagree, but actually clearly we can find Taliana in very highly competitive environment and the population are stable in this highly competitive environment. Okay, so first approaches. So what we wanted to do in this experimental evolution is to monitor the demography and the genomic of experimental population according to intensities of interspecific competition. So the approach that we adopted is quite different from the approach that Magnus proposed yesterday. So you remember Magnus yesterday proposed to use a bunch of several natural ecotypes, like 200 Swedish ecotypes, and to put all these ecotypes together in the same, but maybe and to put them back to the native population. But I was worried with this approach that actually this is what we call, that have been developed, what we call genotype sorting, and especially for it's very well known for selving species. It has been modeled in the 70s that actually it corresponds to genotype sorting. So actually you would select very few genotypes, and actually it's very hard to identify the genetic basis of this. So the approach that we decided to adopt was to cross two populations, so named Beau and Anctu. So these two populations are located around Lille, and you can see that one Beau, actually there is not a lot of competition, interspecific competition with Taliana, and in this population there is a high competition level with Anctu. So we crossed these two accessions. So we got 17 F1s and we got F2, and we set up 100 experimental population that correspond to the cage population that Dmitry Petrov showed yesterday with Drosophila. So you can see here what we call this is our experimental population, the cage population, but we didn't want to be also, we want to be sure that the seeds that we will be follow, that the plant does not correspond to native seeds, come from native seeds. So what we did, we decided to set up the experiment in experimental station at Vimereux, which is close to the English Channel. And actually there was some recording about the plant communities, and there was no Thaliana that have been recorded in this place. And we also test that actually we took some soil that we bring to the brought to the greenhouse back to the greenhouse, and to test whether there was any germination of Thaliana, and we didn't find any germination of Thaliana in this on this soil. Okay. So here, we decided to plant to have two period of sowing, of planting the seeds, so the early May and the mid-June. And actually these 2 days correspond to the early dispersal event of the 2 accessions that we were growing in a common garden, just to be sure to mimic what will happen in natural populations. And then, so we planted the seeds in May and June 2012, and then we're expecting them to germinate like early September, like in the other population. But in 2012, actually, the summer was really rainy in this in the north of France. So actually, we had a first generation during the summer of 2012, and so it was allowing population establishment, and then we have like 3 generations. So here, for example, for the first generation, so what we did for the first date, actually, we monitor the demography, and we also collected plants to estimate to characterize the genomics of these populations. So here the number correspond, because this is a destructive approach. So we have 100 on experimental population, and we're just randomly chosen 3 populations among the 100, and we just actually destroyed this population for the use of the experiment. And so we had the same protocol, like we follow the demography over the life cycle of Thaliana. And in the second generation, so we also characterized the demography on the genomics, and we applied 2 treatments of interspecific competition. So in one treatment, there is no intervention, we just let the other plants to grow. And for the other, we just cut the other plant species than Thaliana in this. So we have a reduced level of competition and normal level of competition. Okay, and here you can see the number of populations that actually we use for each of these sampling date. Okay, so first the result about the result about the demography. So you can see here, so the number of the population size according to the different sampling point during the first generation, the second generation, and the third generation. So you can see actually there was a that the population, the seed germinated very well, but there is a decrease, a slight decrease of the population over the lifetime cycle of Fabrice Thaliana. And then clearly there is a big increase of germination for the second generation. And then at this moment we apply the two treatment for interspecific competition. And you can see that over time, clearly that the size of the population in presence of companion species are reduced compared to the population where actually there is the population size in the treatment where we reduce the presence of companion species are higher than the population size in presence of the companion species. And actually this effect is even more dramatic at the start of the third generation. So we characterize the genomic of this population, of this experimental population at the very beginning of the 3 generation. So here are the results. So what we did first is that we characterized, we seek the DNA of 500 F2 plants that we grew in greenhouse conditions. So we planted 500 seeds, F2 seeds, and we got a germination rate of 97%. And this is what we observed. So here, 50%, this is the expectation across the genome, across the 5 chromosomes of Thaliana to have a 50%. But we know, like in any crosses with between 2 accessions of Thaliana, that there is a distortion of segregation. And this is what we observe here. There is distortion of segregation against the allele of the bow population. So then here, what was, this is the genomic composition of the population we observed in June 2012. So the first, actually the first, at the beginning of the first generation. So clearly we see that there was an increase in the allele frequencies of BO14. And there was like here, what I represent is like when it's negative, this is a decrease in the allele frequency of ANC2. So ANC2 is a population where we have a high level of competitions. So then, so clearly, so between this one was the start of the first generation. Here, this is the start of the second generation, and you can see there is not much going on. So clearly, the population were able to establish, and there is no so much variation in the genomic composition. So now let's see what happened when we apply the competition effect treatments. So first, I will represent when there was reduced level of competition. So yeah, the C minus. So here you can see that there is some selection, even like there is actually not like no interspecific selection. There is a reduced level of competition. And you can see that there is some selection of genes that have been selected, some genomic regions that have been selected, and they all belong, actually come from the ANC2 population. And now I will present the results where we have a big intensity, a big level of interspecific competition. And you can see here there is a lot of genomic regions that have been selected by the presence of interspecific competitions. And you can see, as expected, actually, all the genes, all the genomic regions selected actually come from the population that, from the natural population where we observe highly competitive environment. So yeah, so it seems that the relative fraction of the genomic regions and the selection increase with the level of presence of companion species in very few generations. Here, it's only in between 1 and 2 generations. Okay, so the second approach, this is a resurrection study. So for people who are not familiar with this approach, so what we do is that we collect seeds from the same population, from plants of the same population over several generations. And then when you collect all this, after collecting all these seeds over like several generations, you plant all these seeds in the same environment. And actually, this is really interesting because this type of study can actually distinguish the relative roles of plasticity and the adaptive evolution in phenotypic response. So to set up this resurrection study, so I focus on the population 2A. So this population is from the village Toulon-sur-Arroux, located in the south of Burgundy. So this population is located under this electric fence. This is a 350-meter electric fence separating 2 permanent meadows. So actually, I know very well this population because this population is located on the farm of my parents. So it was pretty easy because I was the only one to be allowed to go to there. So, I collected 80 the seed of 80 accession in 2002, 115 accession in 2007, and 150 accession in 2010. And here you can see the location of the plants from which I collected seeds along the transect. Okay, so I was talking about, spatial heterogeneity for abiotic factors, but also biotic factors. So you can see here, even like for the soil condition, like for the pH, over like here, so this is a transect over from 0 to 300. And you can see like even in less than 50 meters, you have a big variation of the pH. So ranging from 4.9 to 6.1. And yesterday I was talking with Ute about this, and actually she told me that she observed the same kind of big variation at a very small scale in her population of Arbres salary. And we observe the same pattern for other components of the soil, like the nitrogen content and the concentration in aluminum. So you can see that clearly there is big variation even in this one population. So on the plant-plant interaction, you can see here some Thalliana and Thalliana interacting with different companion species, Veronica arbanensis, Serratium glomeratum, but also other grasses, including Poa annua and Festuca rubra. So here, we decided to set up the resurrection study, so to set up an 8th generation. So we only consider the 80 accession from 2002 and the 150 accession from 2010. And we directly planted, so we didn't want to plant the seeds actually in a common garden, like in greenhouse conditions or like in the field yard. We exactly, okay, we want to do the same thing. We want, because of all this ecological variation that we observe within this population, we want to plant the seeds directly in the native habitat of this. And so we decided to focus on 3 different types of soils. So this soil differs according to the pH, the content of the nitrogen, and other parameters. So we have 3 different types of soil, and for each type of soil, we have 2 conditions: absence or presence of Poa. So we have 6 macro habitats, and we planted all these accession in these 6 macro habitats. So this is a 10-month experiment. So when I heard people that saying that actually Italiana is a short life cycle, no, actually, no, go to the nature and you will see that actually it's more like it's a 10 months life cycle. Like I told you, actually this is a family project. So my little brother helped me to prepare the experiment. Then I had the help of 2 PhD students and 1 colleague, Laurent Selem, Etienne Baron, and Romain Beloutrex, to plant all the seeds. I even trained my mother to recognize when the plants were starting to bolt, actually, and she did a very good job, actually, because she was taking pictures. But I think also it was a way for her to call me more often. And so, yeah. And finally, so here, during the last 2 months of the experiment, actually, these have been my best friends, actually, the main social interaction I had during the last 2 months. Okay, so we phenotype 25 traits related to phenology, accumulation of resources, architectural and seed dispersal related traits, but also reproductive traits, the length of the fruits, the number of fruits on the main stem, on primary branches, etc., etc. So 29 traits. Okay, so I will first talk about, present the first results. So here you have the list of the 29 traits. So actually, at this point, we don't care about the name of the identity of the traits. And just don't pay attention to the color. I will go back to that later. So at the very beginning, so what we have like what we call 174 ecophenotypic traits. So what we call ecophenotypic traits is like because we have 29 traits measured in 6 macrohabitats. This is what we call ecophenotypic traits. Out of these 174 ecophenotypic traits, 144 were heritables, with a mean broad sense heritability of 0.7, which is quite high, actually, and the range also equals high. We can go up to 0.87. But what is interesting to see, so like actually in the gray box actually correspond to non-heritable traits. And you can see that depending on the macrohabitats, that actually you will see that some traits could be or not heritable. For example, I can take like this, where is this? Yeah, I cannot see myself, the rosa diameter. Yeah, the rosa diameter actually is not heritable in the first macrohabitat and the 5th macrohabitat, but is heritable in the other. So clearly, depending on where you will, on the ecological conditions, the native ecological conditions, some trait could be heritable or not. Then now we will pay attention to the colors. So the color represents the rate of evolutionary change calculated with in aldans. And so in red, pink and red, it means that there is an evolution for bigger values. And in blue, it means that there is an evolution in smaller values. So actually, we find that with 70, among all these heritable traits, 76 evolved, and 68 did not evolve. We don't find them to evolve. But again, the percentage of phenotypic traits, ecophenotypic traits that evolve clearly depend on the macrohabitat. So here, this is just the soil C, in absence or presence of Poa annua, and you can see that clearly it dramatically changed actually what you can say that there is only, here for example in this one, we find that there is only 23% of the traits, heritable traits that evolved, but in this one is 57. So even when you want to work on phenotypic evolution, it's really important to maybe to do the experiment in several experimental conditions. So here, this is just an example for 2 very key traits. So for the bolting time, so from the germination on the onset on flowering, and you can see that there is an evolution from 2002 and 2010. We observe actually the same evolution, so an evolution to more late flowering plants. So there is an evolution to more late flowering plants. And we also have this trait, so the total seed production on the main stem of the plants. And we also observe that there is an evolution to produce more seeds on the main stem. This is true for 4 out of the 6 macrohabitats. But the question, but what was interesting is that for this, actually, we have the same phenotypic evolution, but actually for flowering time, the accession that flowers the latest actually are not the same. So you have exactly the same, you observe the same phenotypic evolution, but actually the ranking of the accession are totally different due to G/E pair interactions. So actually the accession that flowers the latest in the macrohabitat A are not the same that the accessions that flowers the latest in macrohabitat B. So same phenotypic evolution, but not actually the same accession close to the global phenotypic optimum. So, and you can see, for example, here, this is particularly evidence for the total seed production on the main stem. You can see here, for example, that there is no lot of G/G interaction in this case. Okay, so the question that we have, whether can a similar phenotypic evolution be driven by distinct genetic bases across macro habitats? So we sequence all this population, all these accessions, the 195 accession, and we also sequence other accession located in other population located very nearby the 2A population, and you can see that there is a big overlap between the accession from 2002 and 2010, and they are distinct from this other local population. And we also observe a very short LD in this one. So here, this is a GWAS that we perform on the bolting time. Here you have the Manhattan plot across the 5 genome of the 5 chromosomes of Taliana, and in red, the red SNPs correspond to the top 200 SNPs. And actually, there was no shared top SNPs, actually, among the 6 environments, and 88% of the top SNPs are environment-specific. So I'm running out of time, right? Yes? Yeah, okay. So, but now if we consider like working with multiple traits at the same time, so with at the multiple time, because here I was just focusing on flowering time, and we can see that actually clearly we have different genetic bases involved in the phenotypic evolution. But now if we look at the multiple traits at the same time, so here we look at the effective number of ecophenotype, and we observe a L-shaped distribution of the effective number of ecophenotype, and actually this is in line with what people observe in nematodes, yeast, and mice. We also observe, so I will skip that. So we also observe that there is a synergistic pleiotropy, so the total effect size increase with effective number of ecophenotype faster than linearly. So actually, so there is clearly a synergistic pleiotropy. And I think I will not have the time to develop this, but actually we demonstrated that there was a selection on intermediate degrees of pleiotropy, so which was in line with what was predicted by theoretical model. So clearly, so what in this kind of experiment, in the native habitats, we can show that depending on the native environmental conditions, we can have the same phenotypic population. So it seems that you can have the same phenotypic evolution for the population to reach the same new phenotypic optimum, but by different genetic solutions. So this kind of genetic association should be able to maintain the genetic variation within the population. And this is what we observe, actually. We don't have no loss of genetic variation in this population, but there is an adaptation. And finally, last slide. So this is something that we started in the team like 9 years ago. So it's to do a demographic, phenotypic, and genomic monitoring of natural populations. So now, we identify 168 local populations in the southwest of France. We characterize them in 2014, 2015 at the genomic level, for the climate, for the soil physicochemical property, for the microbiota in the soil, the root, and the leaf for the plant community, but also the degree of the level of urbanization. And now what we do is to follow all this population every year, except like in 2010, so to estimate the population size and to phenotype more than 30 phenotypic traits. And actually, this is quite amazing to see that with some populations, they are very stable at the demographic and phenotypic level. Some others, there is clearly demographic disturbance, and actually we have very different conditions, and we hope that actually, because at the end, in 2026, we want to do exactly the same characterization and to see how all these demography and ecological factors affect the genomic evolution. And like this, I want to finish and thank the finance, my team. You can see here on all the financing institute. Thank you very much.